This chapter we talk about Mendel and Mendelian genetics. And Mendel was a young man who, because of his place in life, he was a poor guy, and because of his place in life, he was sent to school. Well, I mean, he got to go to school, which is actually kind of a good thing. And we're talking about the 1850s. Anyway, to make a long story short, he ended up entering a monastery, and that monastery actually allowed him to study in Vienna. Um, I believe this is the way the story goes. I may be getting my timeline messed up. But anyway, he studied physics and chemistry. And so he actually came from an agricultural background. So what he was able to put together was that agricultural background with the foundations of what that time it was a very active scientific experimental world, or that was discovering quite an enormous amount of things. But anyway, he worked with peas, garden peas, and there are many reasons why he worked with garden peas. First of all, they have a short lifespan, and so generation after generation, they were able to, they made good experimental subjects that way. And they also you're able to actually make sure you guarantee the breeding of them. So he worked with about 5,000 pea plants and that was the agricultural side of himself. And the science side of himself is he made very, very extensive notes and kept great books and results of those up to 5,000 plants. And so the upshot is, and he wasn't particularly considered, he wasn't given any accolades in his lifetime, but we consider him now the father of modern genetics because of his experiments with his garden peas and the fact that he looked at them in a very scientific way. So as I said, the advantage of peas for genetic study is there are many varieties with very distinct features, such as flower color or whether the pea pods are wrinkled or not, and their height, etc. Most of his experiments worked with flower colors, although he has there are about 6 different traits that he looked at. The mating can be very much controlled because you can brush the pollen on. Each flower has a sperm producing organ and an egg producing organ, and that cross pollination can be very very well controlled by dusting pollen from one to the other. And then the other thing that he did, he chose characteristics that occurred in 2 alternate forms. And so we're looking at purple flowers and white flowers. And you can think of characteristics as traits, or the idea that we have with regard to inheritable traits or characteristics. And he also used true breeding plants. And what we mean by true breeding plants is plants that no matter how many generations you breed them, they always come up in a very predictable way. Now, they're not always coming up the same, but in a very predictable way with regard to purple flowers and white flowers. So that he had an idea, and one of the reasons why in your head you might be saying, why is all this important? Because what he did, the science part, is he figured out mathematically what the probability would be, or what the outcome would be of a particular breeding pair of peas. And that was some pretty sophisticated math at that time. It's kind of sophisticated math at this time. A typical experiment, Mendel would mate 2 true breeding varieties And that process is called hybridization. And just a reminder, remember so much of what this particular course is learning what words mean. So we use that word hybridization, but what it means is that you're actually breeding, and we think of it particularly with plants, that you have an idea, you being the person who's actually creating this plant, has an idea of what you're going to end up with. And so the parents are considered the P generation. And then hybrid offspring of the P generation, so the offspring is the F generation, the familial generation. That's Latin for son. And then the offspring of the F1 generation is the F2 generation. And generally what happens is it's the F2 generation that's looked at in numbers. So what you have in this particular graphic is down to the F2 generation, he mathematically counted the colors, and we're talking about large varieties of test groups. So 705 purple flowers, 224 white flowers. So what you actually have is a 3-to-1 ratio about a 3-to-1 ratio with regard to purple to white flowers. And he consistently realized that this was the case. As long as he was working with these true breeding plants, consistently worked with, realized that these were the numbers you would come up with. He considered, because of that, that the purple flower color was dominant. He certainly had the greater number of them. Now please understand, these terms are not attributed to Mendel, but he conceptually thought of it as dominant. These actual coining these terms certainly were I guess were certainly with Mendel in mind. So this dominant and recessive concept came out of these experiments. And then Mendel did develop this hypothesis to explain a 3-to-1 inheritance pattern that was consistently observed in the F2 offspring. Like I said, the 700 plus and the 200 plus, 700 plus purple, 200 plus white give you a 3-to-1 inheritance pattern. Because of these experiments, he came up with certain theories and laws. So 4 related concepts make up his model, and these concepts then were built upon and became the foundation for what we know about genes and chromosomes at this time. Really what he was talking about are dominant and recessive genes, and the colors then became what's known as an allele, or a locus on that particular dominant or recessive gene. The first alternative versions of a gene account for variations in inheritance. And so what happens is, for example, gene flower exists in 2 versions. Then what word we're adding to this is that these alternative versions are now called alleles. So the alternative versions are what account for the fact that you have a little bit of variety in what is seen in the P's, and we now call these alleles. And the genes then reside at a specific place on a specific chromosome. That he didn't know, but certainly this information helped people build on this. And what you have here are the 7 different characteristics of plants, of the pea plants, that he looked at all of these characteristics and also figured out the ratios of each, what the ratio of each would be based on just counting. Then second, for each character, an organism inherits two alleles, one from each parent, male and female, and remember this is sexual reproduction we're talking about, and they occur at the same spot on each of those. At that time it's chromosomes, but at the time they didn't know that the two alleles at a particular locus may be identical, as in true breeding plants, or the two alleles can differ. So you have these alleles showing up on locus of the two chromosomes. They can be exactly the same, or they can be two different ones. And this concept of the same or difference is where we get these terms homozygous or heterozygous. The third, the two alleles at the locus differ, then one is the dominant and one is the recessive. And the dominant one is going to determine what is expressed, what that particular plant looks like. So if you have two different ones, the dominant one will determine the appearance. The recessive one, one won't know what the recessive one is unless you go through these breeding experiments. The F1 plant had purple flowers because the allele for that trait's dominant. And these are his fourth observances, I guess, which now is known as the law of segregation. The two alleles for inheritable characteristics separate during gamete formation. So they one will go to the remember, just a little bit of review of meiosis is you start out with a precursor cell. That precursor cell goes through two phases of division. After the first phase, there is no replication, so when it gets down to the final division, you have a half complement of the genetic material, and so that half complement from the biological mother gets together with the half complement of the biological father. And what he's saying is that from the maternal side and from the paternal side, you will have a different allele from each. So the same locus and the same inheritable trait, but one will come from the mother, one from the father. And if in fact there's a dominant one, that one is the one that's going to be expressed. The segregation of the alleles corresponds to the distribution of homologous chromosomes. I knew I could get it out. Two different gametes in meiosis. So homologous meaning same chromosomes. The chromosomes are the same, but they can have different alleles. Organism with two identical alleles is said to be homozygous for that gene. If the two alleles on the two chromosomes that are exactly the same, it's a homozygous for that particular gene, just for that gene. Not the chromosomes aren't homozygous, but for that gene they are. The organism that has two different alleles is called heterozygous for that particular gene that controls that character. And unlike homozygotes, heterozygotes are not true breeding. So every single one of the plants that he used in his experiment were homozygote, meaning they had the same trait on each of the chromosomes. We'll get started on this one, and then we're running out of time, but because of different effects of dominant and recessive alleles, the recessive one is not always revealed in its composition. So one doesn't see the recessive part if it's not expressed, and we'll pick this up on the next one.